Hey, honors biology students, this is the last video of notes over organic compounds, and it's over proteins, so we'll go ahead and get started. Proteins are a chain of molecules called amino acids. There are 20 different naturally occurring amino acids. They build muscle, skin, ligaments, bones, hair, lots of things that are tough connective tissue. Those all seem pretty tough, and proteins make up all of those. The muscle part you know, but you may be a little surprised by the skin, your ligaments, your bones, your hair, all made of protein. Proteins are made of carbon, hydrogen, oxygen, and nitrogen. Some have sulfur and phosphorus and other things. This is a basic structure of a protein. It's called an amino acid. And proteins are chains of amino acids called polypeptides. So your basic group, as far as your amino acid, is going to be constructed. Your basic structure is around a central carbon. You have an amine group, and that's an NH2. You have a carboxyl group right here. And you have an R group that is over here. Now, R is not an actual element. R is representative of some group that you have. So those are the things that are around this carbon. This H actually goes up at the top here, but they just put it right next to it. I guess they're friendly. So this is your basic structure for an amino acid. The amine group, the carboxyl group, and then the R group. There are 3 parts of an amino acid. So here are some real amino acids. Let's circle the R group, star the amine group, and put a square around the carboxyl group. So do this in your notes with me. The R group, actually let's start with the amine group, okay? The amine group is the NH2. So we're supposed to star it. So do you see the NH2 in glycine? Go ahead and put a star right here. In alanine, another amino acid, it's right here. You can put a star by the NH2. That's the amine group. And then valine, you can put a star by that one. So amine just means it has nitrogen and hydrogen in it. So those are the stars for the amine group. Let's do the carboxyl group. Let's put a box around the carboxyl group. So that's COOH. Now up here, we have a C double bonded to an O and then an OH coming off of it. That's COOH. Down here, they just wrote it out, COOH. They didn't show the bonds like they did up here, but it's the same thing. So let's put a box around the carboxyl group. So box that guy, box that carboxyl, and box that carboxyl. So we have amine group, our carboxyl group, and then the last group is the R group. We're supposed to circle the R group. So in glycine, the R group is the hydrogen over here. This hydrogen for carbon is just like the packing peanuts extra hydrogen that is a filler. But glycine just has a hydrogen as its R group, so circle that. In alanine, in the amino acid alanine, the R group is CH3, so go ahead and circle the CH3. In valine, the R group is this whole piece here, the 2 CH3s connected to the CH. So the R group changes with every amino acid. The R group makes the amino acid different. But the actual amino acid structure is constant, meaning there's a central carbon, there's an amine group, and then there's a carboxyl group. And finally, there's an R group, and those are all different in each amino acid. So I hope that helps kind of explain how amino acids are different. And remember, amino acids make up proteins. Okay, now let's look and see how we can put amino acids together. Amino acids can be linked together in long polymer chains to form protein molecules. They are joined together by what we call peptide bonds. A peptide bond is when a carbon and a nitrogen bond together. So you can write a C, bond N, and then you have a peptide bond. So let's take a look. You will notice from the diagram that a water molecule is formed when two amino acids are joined. What kind of reaction is this? So here's the first amino acid. Let's do a little bit of practice in identifying the parts of it. Here's the central carbon. Now this one looks a little differently from the last slide. The only difference is that the amine group, the NH2 group, is over here. That's just, they just wrote it over here instead of down here. The carboxyl group is right here, COOH. Let's look over here. We have the carboxyl over here, the amine group here, and they just said R1 and R2 because they're not doing specific amino acids. They're just saying here's the first one and here's the second one. So we have the three groups, the carboxyl group, the amine group, and the R group for both amino acids. What we're trying to do is put them together and synthesize them and make a peptide bond where this carbon is bonded to this nitrogen. Well, there's some stuff in the way. It looks like there's an HOH in the way, and if we can get rid of it, we can make a peptide bond where this carbon is actually attached to this nitrogen. And now we have this one amino acid connected to the next one. And what do we lose? We lose water. And so this reaction is called dehydration synthesis as well. Before we had a glucose and a glucose bonded together and gave water off. Now, when we make proteins, we are making peptide bonds with carbon and nitrogen, and we'll make what's called a dipeptide. When two amino acids join up, they make up what's called a dipeptide. So di also means two, mono, di, tri, 1, 2, 3. So a dipeptide is made of two amino acids. More and more amino acids can join together to form polypeptide chains, which may be from around 10 to many thousands of amino acids. A polypeptide can then fold or coil or become associated with other polypeptides to form a protein. So let's continue. This is the reaction that bonds two amino acids together is called dehydration synthesis. Water gets released, and this is called a peptide bond. All right, protein structures. You have pictures here of 4 different protein structures. So just write down in your notes the yellow. So when we do protein structure, we have 4 different structures. So the first one is the primary structure. A primary structure is simply a list and order of the amino acids. We just put amino acids together, and we did that by dehydration synthesis. And when you just have amino acids in single file connected to each other, we call that its primary structure. So again, just write what's in yellow next to the first bullet of primary structure. And you have this picture too. This is just a sequence of amino acids. The secondary structure is the 3-dimensional arrangement of the polypeptide chain. So it actually begins to twist. It has some kind of 3-dimensional form to it, but it's pretty simple. It might be in sheets, and it might be in some kind of helix, an alpha helix perhaps, and turn this way. It has a little spiral coil, so picture it kind of like a spiral staircase, or just like a twisted up, just a line that's been twisted up. In other proteins, polypeptides can fold into sheets. Sometimes there's no regular secondary structure, and the polypeptides form a coil. How are the amino acids attached? By hydrogen bonds. So they're actually attached here longitudinally, as well as attached here by peptide bonds. They're attached by hydrogen bonds here. So to apply that, it would be like this alanine and lysine would be attached together by hydrogen bonds. This lysine and alanine, they would all be kind of attached together long ways. Even though you can't see those, hydrogen would be attracting those together, and that's how they coil up. So that secondary structure is a three-dimensional arrangement. The tertiary structure, that is 1, 2, 3, third degree or tertiary structure, folds those sheets and helices into shapes. So it's like taking the secondary structure, as you can see it kind of in the picture, and folding it into another shape. And it has very irregular shapes. They're not symmetrical. This is an example of one. And basically they begin to get kind of globby. So the three-dimensional shapes are held together also by hydrogen bonds, and that's tertiary. The last structure is quaternary structure. Quaternary structure of a protein takes the tertiary structures, as you can see this one was maybe in the last one, and then puts them together in a quaternary structure. So it's very complex. Enzymes are most often quaternary structures, and other big proteins are quaternary structures. So there are a couple of types of proteins. There are fibrous proteins and globular proteins. So let's look at those. Fibrous proteins do not have tertiary structures. They have primary or secondary. So they're just these long, just spiral, twisted chains of amino acids. They are insoluble in water and very tough. Example of fibrous proteins: connective tissue, tendons, muscle, silk, hair, nails, horns, and feathers. Just some examples of fibrous proteins in living things. So you might take like a secondary structure and then coil it around another one and another one, and they get very tough, and they're not soluble in water, as most of these things are very tough and insoluble. Globular proteins are the second type. So if it's not fibrous, it's globular. And globular proteins are a big glob, exactly the way it says. They have complex tertiary and sometimes quaternary structures. So all these tiny little balls are amino acids. So they're very complex. They're huge structures, and they are folded into shapes in big globs. Antibodies in your blood, hemoglobin. This is actually hemoglobin, a digital interpretation of what hemoglobin looks like. Hemoglobin is the protein that actually takes oxygen to your cells in your blood. Hormones are globular proteins and enzymes, which I will talk about in a minute. Because globular proteins have very complex shapes, this makes them ideal for being enzymes. So I'm going to stop this video and probably make a different one of enzymes, because enzymes is the last topic, but I'm going to need a few minutes for those. So this is just proteins video, and then I will probably make a different one for enzymes, and then I will tag on nucleic acids to the end of that.